Names Rastina.
Yes. Dear 9th class students, welcome to the class. Morning and good afternoon, everyone. So, today we are going to learn about endoplasmic reticulum. So, KTS Macharla group. So, please concentrate in the class. Okay, this is a NEET class which is different from the state syllabus, and you have to keep keen interest in the class. So, we have completed mitochondria and as well as ribosome. So now our topic is endoplasmic reticulum. Endoplasmic reticulum, we shortly term it as ER. ER. Okay. So in the year 1945, the scientist named Keith R. Potter. The name is Keith R. Potter. He observed that the extensions are coming towards the nuclear envelope from here. So these are what? Outer layers from the cell membrane. You see, this is the cell membrane. This is the cell membrane. From here, what? The tubes or sac-like structures. They are what? Tubes or sac-like extensions. Tubes or sac-like extensions. Tube or sac-like extensions. So, what is the main function? Transfer of message or information or any substances. Transfer of message or any substances from one place of the cell to the another place, or from one cell to the another cell. So endoplasmic reticulum in 1945, it is discovered by Keith R. Potter. So how they look like? They are the membranous extensions. They are the membranous extensions coming from the cell membrane and also from the nuclear membrane. So how they look like? Tubes or sacs? Tubes or sacs. What is the function of this endoplasmic reticulum? They transfer message from one place to the other place, or from one cell organelle to the endoplasmic reticulum. Exactly. Right. So endoplasmic reticulum is of two types, isn't it? Endoplasmic reticulum, it is of two types. One is rough endoplasmic reticulum and another one is smooth endoplasmic reticulum. So RER, rough endoplasmic reticulum. SER, smooth endoplasmic reticulum. Rough means what are present? Round structures. What are those? Ribosomes. Ribosomes. R. So R means what? So if I say RRR, you are going to somewhere. So rough endoplasmic reticulum. R stands for small round-like structures. Very small round-like structures on the surface of endoplasmic reticulum. What are those structures? Ribosomes. What are those structures? Ribosomes. So then that ER is known as what? RER. So what is the main function? They synthesis proteins. They synthesis proteins. So generally we eat food, right? If we eat food only, we do any work. Same like that. Our body is made up of many cells. That cells also need somewhat food. Okay. So where that food is prepared by what? RER. For what? Proteins. They prepare what? Proteins. And that proteins are taken from RER to various places. And smooth endoplasmic reticulum means no ribosomes. Nothing is present. Totally smooth. Totally smooth. They prepare what? Lipids and as well as fats. Lipids and as well as fats. Means endoplasmic reticulum is itself, it is involving in the process of production of proteins and fats. Okay, only endoplasmic reticulum is involving in the proteins and the fats. You all must know that proteins are for what? Growth and repair. Fats for what? They will give instant energy. And these proteins and fats, what they do means if the cell membrane is getting weak or rupture, then they will transfer. This endoplasmic reticulum will transfer the synthesized proteins and fats towards the cell membrane. Understand? If the cell membrane is getting weak, okay, day by day what we become weak, no? So same like that, the cell membrane also will become weak. So at that time, what the ER will do? It will send proteins and fats here. It will become again repair. And also these proteins are sent towards nucleus. Where the proteins are sent? Nucleus. Nucleus contains what? DNA. DNA is made up of what? Proteins. Okay. So once again I am repeating. We are similar like that of our parents or grandparents or our cousins or aunts or uncles. That is because of what? Genes. That is because of what? Genes. Where the genes are present? In the chromosomes. Where chromosomes are present? In the nucleus. So what is the composition of that nucleus? They are mainly made up of what? Proteins. Proteins are what? Living material or non-living material? Non-living material. We are living material, but we are composed of non-living materials. So proteins. Nucleus contains what? Chromosomes. Chromosomes contains what? Genes, which are responsible for what? Heredity. Okay, which are responsible for what? Heredity. For example, human beings, they give birth to human beings only, not dogs or cats. That is because of what? Chromosomes, DNA. Where they are present? In the nucleus. How many nucleus we have? Only one. How many nucleus we have? Only one. So that nucleus is made up of what? Proteins. That is why endoplasmic reticulum is forming what? Proteins. That is built up in what? DNA. That is built up in what? DNA. And one cell will give a raise to another cell. At that time, what should happen? It becomes double. In order to become double, it needs extra source. That extra source is prepared by endoplasmic reticulum, proteins and as well as lipids and fats. And next topic. So this smooth endoplasmic reticulum, in mammalian liver cells, the mammals, in mammals, in the liver cells, it removes toxins. Removes toxins. That is the special character. Okay. In all the cells, it performs only one function: preparation of lipids and fats. But in liver cells, what it do? It removes the wastes. It removes the wastes. Please note it down. So next cell organelle is Golgi complex or Golgi apparatus, or sometimes we call it as Golgi body. Camillo Golgi. Camillo Golgi. He is an Italian physician. In the year 1898, Camillo Golgi, Italian physician. He was the person who discovered Golgi complex. That is why the name Golgi apparatus, Golgi complex, or Golgi body, or Golgi sac. The structure is like this: the group of sacs, that is known as cisternae. So wherever you find group of sacs, sac means what? Bag. Sac we call it as what? Bag. Sac means bag. Why we use bag? To pack any material. Okay, bag means what? It will pack any material. So we call in scientific terms sac. What is that? Sac. So sac, if you see any sacs which packs any material, then that are known as cisternae. What is that? Cisternae. Actually, ER will produce proteins, lipids. So before they are transferred to another place, okay, ER it is producing what? Proteins and lipids. So as soon as it prepares lipids and proteins, it not transferred. So where they are stored? They are stored in where in the cisternae of Golgi. Whenever they are needed, they are transferred. So this Golgi complex, it is related with the post box. Do you know about post boxes? Letters are transferred from one place to another place. Transferring of letters from one place to another place, it is resembled with ER. But where they are stored before transferring to other place? In the post boxes. So this Golgi apparatus is, it is compared with what? Post box. It is compared with what? Post boxes. As the post box, it will store no. Where they are needed, they are dispatched. Okay. If they are needed to nucleus, it will be dispatched. If they are needed for lysosomes, dispatched. If they are needed for cell membrane, dispatched. Means what? According to the need, it will dispatch. Before that, it is stored where? In the Golgi complex, in the cells which are known as cisternae. Which are known as what? Cisternae. I again repeat, endoplasmic reticulum will produce lipids and proteins. Okay, that should be transferred to various parts. So in the cell, many parts are present, many organelles are present, but all of them don't need proteins and lipids, right? Where they are needed, they will be transferred. Before transfer, they will be packed by the Golgi complex and transferred to various parts by Golgi complex only. So what is the main function of Golgi complex? It will pack and transfer. It will pack and transfer to various parts. Golgi complex. Please note it down. And next to this, Golgi complex will produce proteoglycans. Proteoglycans. Lysozymes. So generally, in our biology or in any science words, the science words are generally taken from the Greek and Latin words. You will see in your textbooks that science is derived from this word, Cell is derived from this word, Latin and Greek. Because it was known that Greek, you know about Alexander. Alexander is from Greek. He's a great leader. Okay. In those days, during 17th, 16th centuries, the Greek and Latin places are moved as the famous. Okay. They only had made discoveries. They only came friend and made discoveries and made knowledge. So many of our terms which we are using now in the science are mostly derived from Greek and Latin languages. So lyso means break. Lyso means break. Science means body. Okay. So break body. Lysozymes already you have studied. Lysozymes are known as what? Lysozymes or sometimes known as lysosomes. Lysosomes. So soma, you have studied soma. Soma means body. Soma. Body. Soma means what? Body. Okay. Lyso means what? Break. Wherever you see lys or lysis, so you can say that it is break. Okay. So means what? They break their body. Suicidal pacts. You have studied, right? So D. Dewey. 1955. D. Dewey discovered lysosomes. Okay. 1955. D. Dewey, he discovered lysosomes. For this discovery, he was awarded. He was awarded Nobel Prize in 1974. 1974 for his discovery. Okay. So in which field? Nobel Prize in medicine. In medicine. So every year we get Nobel Prize. Not we in the sense the scientists will get Nobel Prize. Okay. Art, literature, medicine, physics, chemistry. Okay. So in various fields they will get Nobel Prize. So in 1974 we got Nobel Prize in which field? Medicine. Because it is related to medical practice now. So that is why he got Nobel Prize in medicine. Why? Because he discovered lysosomes in the year 1955. D. Dewey. So they break themselves. They break themselves. Then energy is needed. So that is why they are known as what? Suicidal bats or suicidal sacs of the cell. Suicidal bats or suicidal bats. So is anybody making them to digest? No. They are doing self. Suicide means what? Making self-death, no? So that process is known as what? Autolysis. Autolysis. Auto means self. Lysis means breakdown. Autotrophs. You have studied no autotrophs. Auto means what? Self. Troph means synthesis. So already I said lysis means what? Break. Auto means what? Self. You all members know autobiography. No, auto means what? Writing themselves. Self. Auto means what here? Self. Lysis, breakdown. They are making self breakdown. That is why the process is known as what? Autolysis. So when they break down water release, hydrolytic enzymes will break down. Hydrolytic enzymes are present in the body. These hydrolytic enzymes only will break down. Enzymes are responsible for the reaction process. So what are present in the body? Hydrolytic enzyme. That hydrolytic enzyme only will break down the lysosomes. So another thing is what? They break down phagocytes. Means foreign materials. If any foreign material enters into the body, no, that lysosomes will break down. Okay, so this is about lysosomes.